Hello, everyone, and thank you for tuning in to another Fluence webinar. My name is Dr. David Hawley. I am the principal scientist here at Fluence, and with me today is Taylor Kirk, our cultivation advisor. Together, Taylor and I work together to execute scientific research and then translate it to commercial application, and that's what we hope to show you today. We're going to walk you through a lot of our dynamic lighting research, specifically focusing on dynamic spectrum, and then we're going to translate the learnings from that research to a product solution that Taylor's going to tell you about at the end of the presentation. Pardon me. Now, if you happened to see my presentation with Cannabis Business Times about 3 or 4 weeks ago, then the first half of this presentation is going to look pretty familiar to you. I would say it won't be a waste of your time to watch again because there is quite a lot of content in this presentation. And if you asked any questions in that presentation that we didn't have a chance to get to, you can ask them again here. Otherwise, I would say, again, just stay tuned to the end because we're going to take those learnings and the science that I talk about in the first 35 minutes or so and translate that to a new product solution that I think a lot of people are going to be really interested in. Okay, and with that, we'll dive in. I want to start this presentation by level setting why any of us care about dynamic lighting. Right now, you may have this idea that today you want to be able to manipulate red or far red or blue or light intensity or any of these things because you want to dynamically drive yield or quality or chemistry or a certain morphological response. But if you extrapolate that thinking forward, you say, well, wouldn't it be great if for a given cultivar, I knew exactly what lighting inputs I needed to do to achieve more myrcene or change the ratio of monoterpenes to sesquiterpenes or to cannabinoids or anything like that? And wouldn't it be great if I also knew what I needed to do with my climate and what I needed to do with my fertigation and have all of these different environmental inputs talk to each other and make tiny little tweaks to their environment to ultimately produce these perfect plants? Wouldn't that be great? The truth is, this is maybe science fiction, and we've probably all seen something like this. If you've seen like Interstellar or Ender's Game or any of these movies or books or stories that talk about this sort of dystopian future, but there's always this amazing garden and this amazing kind of living ecosystem where you have all these perfect plants talking to each other and talking to systems and, outputting these perfect, wonderful things, right? And like all of that, lighting is an important piece of this equation, right? Lighting has the power to be very creative, right? Lighting can drive yield and lighting can steer morphology and it can drive certain chemical profiles like I just talked about. Lighting can also tactfully be used to destroy things you don't want, right? Certain wavelengths of light like UVC can obliterate pathogens, therefore getting you one step closer to a perfect lighting system to give you these perfect plants. But the thing is, lighting isn't free, right? All we are doing is repackaging energy from electricity into photons. So for every benefit that we get from lighting, we need to ask ourselves the question, is this worth it? Right? And theoretically, you would ask this question for how you manage your climate in an automated way and how you manage your fertigation in an automated way. And you would look at all of these different environmental inputs and how they interact, and you'd say, is this worth it? The thing is, this isn't actually as far out into the future as you might think. The idea of automating greenhouses is actually kind of a reality today in 2025 in crops like tomato and cucumber and strawberry and lettuce. These crops are very, very well established in their genetics and in the way that you grow them, right? These are the two pillars that you need to have consistent if you want to have amazing automated systems to give you really perfect, consistent plants. And think about why this is, right? Like if you're growing tomato, probably that means you're growing a cultivar called Marlies. Excuse me. If you're growing strawberry, it probably means you're growing a cultivar called Albion, right? And even if you've never grown a tomato in your life, you can probably go and pick up a greenhouse tomato handbook and it will say, use this plant spacing, use this fertigation media, use this lighting regime. If you're in the Netherlands, do this. If you're in Ontario, do this. If you're in the south of France, do this, right? And yeah, there's something to be said for experience, but as long as you follow that handbook, you're going to get reasonably close to a very good quality crop. Obviously, I'm oversimplifying for the sake of the storytelling here, but you get it, right? And that's the same with strawberry, it's the same with lettuce. The methods and genetics that you use have been established by decades, if not centuries, of academic and field research that have said, they've come together and said, this really is the best way that you do this. And this really is how climate interacts with lighting, interacts with fertigation and transpiration and everything else. If you do this, you will get good plants. When it's that simple, you can actually program and automate that, right? There's a thing called the Autonomous Greenhouse Challenge that Wageningen University does every year. And there are companies like Koidra, like Blue Radix, that have shown repeatedly now that they can out yield growers that have had generations of experience in their life. Now, whether or not those systems are totally efficient at this point is beside the point. But as far as pure productivity goes, the technology is there now. Cannabis has a long, long way to go. So think about this, right? Cannabis doesn't have consistent genetics, and cannabis does not have consistent cultivation practices, right? If I'm driving a cultivar called Blue Dream at a research center down the road at Texas Original Compassionate Cultivation, that Blue Dream was pheno-hunted out of 1,000 or 5,000 seeds that also were genetically diverse, right? And that seed stock was probably very different than a seed stock somebody else has of Blue Dream on the other side of the planet. So even within a single cultivar, Blue Dream, the genetic diversity is so great that being able to consistently predict how that plant's going to respond to an environmental input like light is actually pretty tough. And when you think about the genetic diversity that exists across different cultivars in cannabis, you can kind of throw that out the window. What's more is that you then look at cultivation and say, well, are people at least cultivating the same way? And the answer is no, right? The style of cultivation in North America is very different than in Europe and very different from the rest of the world. What's more is that even within North America, you can look to the West Coast and there's a cultivation style. And then you look at the rest of Canada and the United States and cultivation is a little bit different, right? And that's driven by a lot of things, but primarily regulations and people having to follow certain rules to not get shut down, basically. That means optimizing your cultivation and genetics and having those things be consistent and together is really a different beast than what you would expect in something like tomato and cucumber and lettuce and so on. So again, what does this all mean? How can I say all this and then come to you and say, let me tell you about dynamic lighting in cannabis? Well, the truth is that although there are certain things that are cultivar dependent, where we can say, well, there's this one cultivar over here that responds to far red in a certain way, and we don't know if it's good or bad. It's just interesting. And there's a cultivar over here that we know we can manipulate this ratio of monoterpenes or whatever, but that's kind of just this cultivar. That's not helpful to you. What is helpful to you, I think, are the lighting responses that are conserved across all of the cannabis genetics that we've worked with, which is a pretty broad uh, sort of smorgasbord at this point, for lack of a better term. So what I'm going to do again is I'm going to walk through the different spectral studies that we've done over the last 7 or 8 years and shared with you. I'm going to share with you the conserved conclusions that we see and ultimately conclude with what we think is economically relevant and beneficial to you. So without further ado, let's dive in. Okay, here are the biggest questions that we've asked as it pertains to cannabis spectrum, and ultimately, again, what might inform a dynamic lighting solution of the future. Okay, so starting from the short wavelengths going to the longest, we have things like, can UVB enhance potency? What about blue? It's kind of the same thing as UV, but just a little longer, so maybe it does the same thing. And then we talk about things like red and far red. What if we were to emulate sunrise and sunset by ramping PPFD up and down at the start and end of the day? What about far red? After all, there's far red and sunlight. Maybe it does something. There's certainly this thing called the Emerson Enhancement Effect that far red is implicated in. Maybe we can take advantage of that somehow. And then finally, should I be thinking about PAR or EPAR? Not exactly a spectrum question, but I think relevant to the broader story here and something that won't take long to explain. So we'll look at that. I'm going to start with just what I think are probably the least valuable to you and go on to what I think is most valuable. So I'm going to start with blue light here. And no, I'm sorry, that's a lie. I'm going to start with UV. And again, discuss this idea that applying UVB is going to enhance potency. And again, I'm not going to show a lot of the data in this particular study. If you want to see that, then get in touch with me or anyone on the science team afterward. But we simply don't have time today to dive into the details of all these studies. So I'm just going to tell you the conclusions. What we saw was that when you apply UVB during flower, the more UVB that you use, the worse off the plants were. So you can see our two treatments here, no UV and high UV. Trust me when I say there were a few treatments in between as well. But again, just to illustrate the fact, the more UV you used, the worse off the plants were in terms of yield, morphology, and chemistry, including cannabinoids and terpenes. Everything went down. Now, if we move past that and we say, well, what about blue? Again, like I said, blue is this thing that theoretically could do some of the similar uh, aspirations people have for UVB when it's driving secondary metabolism. But the nice thing about blue is that it's much, much safer for people to work in. And also, you can see when it's on, right? That's a nice thing for a bunch of reasons. And also, the diode technology is much better. Blue diodes are going to last. I can't even tell you how much longer than UV diodes, but suffice to say, UV diodes are not amazing. So, in the pictures that you're seeing here, this is just pictures from one of the studies that we did. In this case, it was a finishing spectrum, and we had the last week or two where we had either just white light or a kind of a 50-50 split between white and blue, and then a treatment that was 100% blue. And by the way, in this particular study, there were not significant differences between the treatments in terms of morphology, yield, chemistry, but it was fun to look at, and I wish that it had driven differences, but it didn't. So why didn't we have differences? And why didn't we have differences with the UV study? Well, I think I want to linger on this just a little bit because there is a perception across the broader world of commercial horticulture that blue light and UV drive plant secondary metabolism. And that's sort of true, but plant secondary metabolism is an enormous umbrella. And the secondary metabolites that blue helps regulate are pretty distantly related to the secondary metabolites that most of us care about in cannabis. So let's just take a look at that. On this next slide, I have a very simplified view of overall plant metabolism, right? So square one, you take some light and you use that light energy to fix CO2 out of the air and some water, and that makes sugar. That sugar is metabolized in the plant cell the same way that sugar is metabolized in our cells. In the case of plants, though, it can make photosynthetic pigments and some other receptors. Or it can be used to make some antioxidants, some pigments, and some compounds implicated in abiotic plant defense. Or it can go this way. You can make some cannabinoids, you can make some terpenes, and you can make some other photosynthetic pigments. Wildly simplified view. So let's just take that one layer deeper. Okay, so like I said, the first step is photosynthesis and making sugar. Specifically, that means the light reactions of photosynthesis, and then the dark reactions of photosynthesis, and then glycolysis, right? That's where you get kind of your main building blocks for all other things in the plant. From glycolysis, you can either go down this path, and you'll notice that there's a bunch of cool things here, but ultimately you make things like chlorophyll and phytochrome. Those are pigments involved in morphological response in photosynthesis. And you can also make ethylene, which is a plant hormone. Or you can go this way. You can consume your sugars and go through the shikimate pathway. You can make things like flavonoids and anthocyanins. Those are like those dark purple pigments that you see in grapes and blueberries. You can also make salicylic acid and auxin, right? Two more plant hormones. Or you can go to the right here. You can go through the DXP pathway, or you can go through the mevalonate pathway. Both of these pathways produce isoprenes, which are 5-carbon chains that you can combine in all kinds of fun ways to make your monoterpenes, your sesquiterpenes, your tetraterpenes. You can make gibberellins, which are yet another plant hormone. You can make carotenoids, which are a secondary photosynthetic pigment. And you can make abscisic acid, which is yet another plant hormone. Okay, so why am I telling you all this? Because blue light only regulates parts of this. Okay, so let's peel away all the noise and just focus on the two paths we actually care about for this conversation. All right, so from glycolysis, you can either go to the left and, oh gosh, sorry, I zoomed in one more time, and I promise it's the last time I zoom in. So right here, I'm showing right down the middle of the screen is the details of glycolysis. You go from glucose, you make glucose 6-phosphate, glyceraldehyde 3-phosphate, on and on until you get to acetyl-CoA. Acetyl-CoA. Tongue-tied. Now, what I want to emphasize here is that there are two very distinct paths that take intermediates from the process of glycolysis and then make secondary metabolites. but not all secondary metabolites are the same thing, right? So if you consume glucose 6-phosphate and phosphenolpyruvate, which you see in blue here, you go to the left, you go through the shikimate pathway and you produce things like anthocyanins. If you take glyceraldehyde 3-phosphate, pyruvate, and acetyl-CoA, you can go to the right and you can produce things like terpenes and cannabinoids. Okay, the way these things are regulated are totally separate. Anthocyanins, we know, is strongly regulated by an enzyme called chalcone synthase. This is an enzyme that needs to be transcribed en masse, and when it has a sort of certain volume, that really helps drive this pathway forward. You get more flavonoids, you get more pigmentation. And if you're a lettuce grower, you love this, right? Because you can get a higher market value for red leaf lettuce than green leaf lettuce. You'll say, great, I'm going to finish my crop with blue light. I'm going to produce more chalcone synthase. I'm going to get more anthocyanins. I'm going to get more money. Amazing. If you're a cannabis grower right now, you're going, oh, that actually doesn't do a whole lot for me. This pathway to the right is what I'm more concerned about. And this pathway is strongly regulated by an enzyme called HMG-CoA reductase, which itself is upregulated in response to mechanical stress like herbivory. So in terms of what realistically is going to potentially change the regulation of these pathways, you are probably going to be better off just throwing a whole bunch of caterpillars into your crop to try and drive that mechanical stress and drive more cannabinoids and terpenes than you are with blue light. Now, I'm not saying you should actually go and do this in your commercial facility, for the record. Nobody go do that and then say, well, Dave from Fluence told you that was a good idea. What I'm saying is that that is an interesting area of research, and I hope somebody does it. In fact, the last time I gave this presentation, someone sent me a link where this is being studied. So more on that later. But the point is, just because blue light can upregulate certain secondary metabolites in certain plants where it does create value, doesn't mean it's going to do the same thing in cannabis. And sure enough, that's what we've seen in our research to date. By the way, all of the research that I'm talking about today is done at PPFDs between 1100 and 1500 micromoles per meter square per second. And that's really important, right? We do our research at those light intensities because we say, well, that's what's commercially relevant. At least in North America, most growers are growing between 1100 and 1500 micromoles per meter square per second. The thing is, there's a significant interaction between light intensity and light spectrum in terms of how plants respond. So, although I am saying one thing about blue light here in the context of the research Fluence has done at these intensities, I want you guys to understand as well that there is other research going on, right? There are a few people that have done research on things like blue or UV at lower intensities at like 400 to 500 micromoles, and they are seeing significant responses that you may want to know about. I think that's great research. I just think it's not as relevant to this audience. And so if you want to learn more about that, I encourage you to read it. And I'm going to be including a link to references at the end of this presentation so that you can go and take a look at that research if you think it may be relevant to you. All right, with that said, I'm going to move on. So blue light, another thing people think it can do is make a more compact canopy. And this is certainly true in a lot of cultivars, and it's true, again, it's one of those light intensity dependent things. But what we've seen in cannabis is that, in fact, the plant is relatively not sensitive to the sort of compaction that you normally get with blue. Even in vegetative development, where we think that blue could be more impactful, what we've seen when we compare a very white light, which contains a relatively high blue fraction, versus something that's more pink with a relatively low blue fraction, the difference in canopy architecture and plant height is pretty small. So that's a morphological response that we really don't spend a lot of time looking at this point. Okay, far red. This one's really fun. Everyone's very, very interested in far red, and there's a whole lot that we have to talk about with far red. I've heard things like far red is great for greater yield. It helps drive the Emerson Enhancement Effect, and it can help put the plants to sleep. There is sort of truth to all of these things. And so I want to talk about it in a kind of controlled way that's most relevant to cannabis, I think. So let me first tell you about the first study that we did with far red. And that was simply to look at whether or not far red really does capitalize on the Emerson Enhancement Effect to give us a photosynthetic response and therefore a yield response greater than if we were just using par light. Okay, so let me talk you through this figure on the left. What you're seeing here is not yield, right? These bars are not describing yield. These bars are describing the light treatments. The first bar on the left that you're seeing is showing that we had, as represented by the blue color, we had 1100 micromoles of PAR light and then 200 micromoles of far red light on top of that. And treatment 2, the next bar over, had the same total flux, right? So instead of a split of 1100 plus 200, it had 1300 micromoles of just PAR light with no far red. And if we were to compare the plant response between those two bars, we can say, well, are we getting some kind of beneficial effect from the far red that we can't get when we apply the same total energy? If we look at the third bar, what you can see here is that we're normalizing on PAR, right? So we have the same flux density of photons between 400 and 700 nanometers, but again, we're adding 200 micromoles of far red on top of that for a total flux closer to 1500 micromoles. And sure enough, that's where we bring in bar 4. If we normalize between treatment 3 and 4, we normalize on EPAR, then we have a total flux between those two of 1500 micromoles. And what we saw was that far red was contributing to photosynthesis, but it wasn't driving yield above and beyond what we were getting with otherwise just PAR light. And there's actually a reason for this. The Emerson Enhancement Effect is a real thing, but it's commonly misunderstood. And so we're going to dive into that just a second and show how and when that may be relevant in commercial horticulture, not just cannabis, but commercial horticulture in general. So, unfortunately, clicking on, let me try one more time. Okay, humor me for a second. We're trying a slightly different thing with how we present this, and it's not behaving itself. So let me see if I can get to this slide and if it animates. Okay, great. So I want to show you the light reactions of photosynthesis and what it may mean for the Emerson Enhancement Effect. So from left to right, here's how this works, right? The first step is that light comes in and accelerates, it excites an antenna complex on photosystem 2 on the thylakoid membrane. When this is excited, that energy is used to essentially liberate an electron from a water molecule with this thing here called the WHCs, the water harvesting complex. And that electron goes on to power a whole bunch of molecular machines to do cool things. So you can see here it gets passed through this electron transport chain from photosystem 2 through plastoquinone, through cryptochrome, through plastocyanin. And I hope you noticed that as it passed through plastoquinone right here, plastoquinone pumped a hydrogen proton from the outside of the thylakoid membrane to here in the lumen. Okay, now that electron in its journey has made it here to photosystem 1. And at photosystem 1, that electron can carry on with its journey. But there's an interesting thing that happens right here. Photosystem 1 can be preferentially energized by far red light. So it doesn't need to be, but it can be. So let's show that. Far red light excites photosystem 1, and that electron is passed up through this enzyme called ferredoxin NADP plus reductase, and it's consumed in the creation of NADPH, which is an energetic molecule. Meanwhile, we have two hydrogen protons down here in our lumen, and they don't want to be in there. There's a biochemical gradient that is encouraging those hydrogen protons to flow out of the thylakoid. When that happens, they pass through another enzyme called ATP synthase, and that drives the synthesis of two molecules of ATP. Now, the thing is, this whole process works pretty well. And this step right here, where far red excites photosystem 1 preferentially, that's great, but unnecessary. That electron will flow through and drive NADPH and ATP synthesis, regardless of if you have far red light. So really, far red only matters if this step right here becomes a bottleneck, right? So if you are in a situation where you're putting a lot of energy into the first half of this process through photosystem 2, through the electron transport chain, and you're getting essentially an energetic backlog right here, at that point, if you were just to add more PAR light and drive this first half even harder, then yeah, things are going to slow down. And at that point, adding more PAR light doesn't do anything. But if you were to add far red and release this bottleneck to help things flow through here a little bit better, At that point, you are experiencing the Emerson Enhancement Effect. You're experiencing a situation where adding far red gives you a net photosynthetic rate greater than what you would achieve with par light. Okay, and with that, I'm going to jump back to this slide and we can carry on with the presentation. Okay, so the next thing we looked at is phytochrome reversion driven by end of day far red. Okay, so far red and red are both received in the plant by a photoreceptor called phytochrome, and the conformational state of phytochrome, literally the shape that it's in, whether it's in like a horseshoe shape or whether it's more kind of a linear structure, that's driven by whether or not it's energized by red light. When it's not energized by red light, it's still active. It's just active in a different way. It does different stuff. And it becomes de-energized either by being in the dark or by being exposed to far red light. And when it's in the dark or exposed to far red long enough, that ultimately, through a whole bunch of signaling cascades, leads to flowering induction. So this actually is a true thing that works in certain species, and it stands to reason that maybe it works in cannabis. By the way, one of the references I'm going to include at the end of this presentation is by someone named Sam Hayden, who recently published research showing that far red is indeed implicated in the biochemical regulation of cannabis flowering. So even though what I'm going to tell you here is that we didn't see significant changes in flowering or yield with our research, the fact is the underlying biochemical mechanisms are there, and so I think it's worth further exploration. Um, anyway, what I want to tell you about our research is that when we tried a treatment where we sunsetted, we emulated sunset by, in the last hour of our day, dipping down par and specifically red light and ramping up far red. What we saw was that in one cultivar, that actually did something. It significantly increased yield, but at a cost. In this one cultivar where we had a significant increase in yield, we had a proportional significant decrease in cannabinoids. So that's the sort of thing where we say, great, yeah, sure, it was only one cultivar out of 7, let's say, but still, that is a response and it's worthy of further investigation. It's not something that we're going to encourage people to do commercially yet because it comes at the cost of cannabinoids and that's no good. But again, with further research, we may find a solution where it's like, oh, well, with the right combination of far red and maybe a little bit of blue or UV or green or whatever the case may be, or maybe something with timing, maybe there's a way that we can get this yield enhancement while also maintaining or improving upon our cannabinoid profile. Okay, red light. If you've been following Fluence's research over the last couple of years, you probably know what I'm going to say, especially when you look at this picture. Red photons are commercially important, right? Red photons are cheap to produce. And if they're cheap to produce, that means that when you have thousands of light with a lot of red photons in your commercial operation, it means your OpEx for your lighting is relatively cheap. That's why when you drive around and you see greenhouses that are commercially lit for whatever crop, you see these generally pink lights, right? It's not because pink is best for those particular crops. It's because red is cheap to produce. And you want to find the lighting solution that can use as much red as possible without negatively impacting yield or quality. The thing is, in cannabis, when you use too much red, right, the absolute dose of red photons is too high, you get something like the picture on the left here. You get what we call photobleaching or white tips. Truthfully, call it whatever you want. We don't know the underlying mechanisms just yet of biochemically what's going on and why you get photobleaching. So one name's as good as another for now. Although we've got some theories that we're continuing to explore, and hopefully in the next year or two we can share more about that. The thing with white tips or photo bleached bud is that it fetches a lower market value. So even though you want to use a lot of red because it lowers your OpEx, if it significantly degrades the quality of your flower and the dollar that you're getting, there's an economic analysis to do there that probably says you got to be really careful not to use too much red. The good news, though, is that again we have figured it out, sort of. We figured out something that I think is going to be very compelling to a lot of people on this webinar. So at this point, we know that it is about the absolute red dose of photons rather than the red percent. But we also know, by the way, that far red amplifies the system. So if you were thinking, maybe far red could help this, it doesn't. But we have found a solution at this point that if you apply red at the right time and you don't apply it at the wrong time, you can significantly save a lot of money without inducing photo bleaching. And what we're seeing is that on average, it translates to about a 19% savings in OPEX. So again, I want to be clear on how I'm saying that. I'm not saying up to a 19% savings, although actually, as I think about it, maybe that is what I said. That's not what I meant. What I meant was that we see on average a savings of 19%. So what that means is if you have a cultivar that is more sensitive to red light, then that means that in a dynamic schedule where you use red at certain times and not others, you're probably going to have to use less red on those cultivars. And your savings won't be as pronounced. Conversely, you're going to have cultivars that are really not so sensitive to red, and you can use more red more frequently, and your savings will be even more pronounced. But again, on average, not up to, but on average, you will experience a 19% OPEX savings. And with that, I'm going to just recap our research on different parts of the spectrum, then hand it off to Taylor to talk about a new product that's going to help enable these savings. So to recap, UV, as far as that we can see right now, really only damages plants. Early applications of UV may induce systemic changes that led to greater yields, and that's something that, again, we intend to explore in the days ahead. We think there is potential for UV. We just don't think anyone has found a commercially viable solution yet. Blue doesn't upregulate all secondary metabolites, but may have some utility in pigmented cultivars. So just like I described with lettuce and a finishing spectrum for lettuce and how you get more anthocyanin, maybe there's a place for that in cannabis too, for like purple and pink flowers. And that's something that we are working on, but don't have a solution for just yet because we haven't seen any cases where that actually works just yet, and we'll keep you posted. Far red, when applied during the photo period throughout flowering, doesn't enhance yield. Unfortunately, it would be awesome if it did, but it just doesn't seem to do it. Emulating sunset with far red can increase yield in certain cultivars, but it comes to the detriment of THC. Far red doesn't help overcome photo bleaching. It actually seems to make it a little bit worse. Red photons induce white tips or photo bleaching when applied with too great of a flux during flower, particularly at times of development when the plant is most sensitive to it. And finally, the big takeaway and Fluence's position on dynamic lighting these days is that red can be strategically dosed throughout the cultivation cycle to maximize yield and minimize operational cost, thereby maximizing your bottom line. So to kind of bring it home to the story that I started this presentation with, there is a path to intelligent cannabis lighting cultivation. And what I would say right now is that there are a lot of things entering the market that have some pretty bold claims about what is or is not possible with dynamic lighting. And I would just say, focus on what's real and what's work and what works. Follow the science. I think the fact that you're here today shows that you're already stepping in that direction. So I thank you for your attention. And with that, I'm going to stop talking and hand it off to my colleague, Taylor Kirk, to talk about our new product solutions. Actually, just before I do that, though, I'm going to show this last slide. Again, screen grab this if you're interested in research outside of Fluence's research. But like I said, there's a lot of great research out there that's just a little bit less relevant, in my opinion, to commercial cannabis production, but nonetheless may be relevant to you depending on what your goals are and how your operation works. And with that, I really will pass it on to Taylor. So Taylor, take it away. And thanks everyone for your time.
Awesome. Thanks, Dave. Thanks everyone for joining us today. And I know that was a lot to digest. And if you did sit in on the Cannabis Business Times version, I know I needed a refresher. So, and I'm much more closer to it than a lot of you are. So, anyways, yeah. So where are we at today? Fluence has pretty much always focused on static spectrum offerings through our product portfolio that have been from tried and true spectrum through for different applications. So we do offer most of you are familiar with our broad white BW4 spectrum, which is really good for all phases of cultivation, propagation through flowering. But we also have offered high red content fixtures for mostly greenhouse applications is how they've been sold and applied to date. But now enter our second gen RAPTR. So RAPTR is a, we hit the market with RAPTR 1 as a one-to-one replacement for HPS, making retrofitting extremely simple and economical for you because there's an ROI value with the additional light you get and the yields you can expect when you manage the environment properly. Um, so we've had great success with that fixture. And, as we went through this presentation, we've done a lot of research on different dynamic strategies. We haven't really come up with something that made a lot of sense to develop a multi-channel fixture with blue UV, red, far red yet. So enter RAPTR 2, which is the first offering that we're going to come out with a dynamic tunable spectrum fixture. RAPTR 2 is going to be offered in what we call T48, which is going to have a 40 to 80% red, two channels, a white and a red channel, and the ability to effectively control the red content 40 to 80%. It's going to come with a integrated wireless system, which is also enabling growers and other dynamic features of rezoning rooms on the fly. Maybe you're going to load new genetics one cycle or at your next cycle, and you want to treat a certain zone, a certain light recipe. It's going to offer that flexibility. And then also we came out with the quad beam 130 to 150 degree light distribution optic. It's going to really maximize uniformity in the grow. So you can see that kind of pictated here on the kind of square throw in all four directions from the fixture, as opposed to like a circular or oval type throw that you're used to from a 120 degree dispersion, or like your historical HPS, hooded HPS optic. What else? Yeah, we've got a new polymer as well coating the LEDs. It's silicone based as opposed to polycarbonate, so it'll last much longer. It won't be subjected to the browning that you often see from blue LEDs over time. Overall, it's just a much more robust fixture with integrated power supply and heat sink with vent down the middle and on the sides. Really sleek, heavy duty fixture. As you can see, it's offered in three different spectrum, the static BW4 and BW4A, but for the purposes of this webinar, we're focusing on that T48, again, the dynamic white and red, which is really going to offer growers the most economical fixture to date from Fluence to be able to manage light recipes based on stage of growth for energy efficiency, maximum output. So I mentioned the wireless control. So we have integrated wireless ability, which is going to save money on installation costs from electrical, no electrical wiring needed anymore. We are able to integrate with something like TrollMaster or a simple controller that Fluence offers, the Shift, which has two-zone control, which would allow us to be a very basic control of this T48 fixture. It also comes with a mobile app feature for adjusting your zoning. As I mentioned, the wireless controllability of rezoning your fixtures on the fly through our mobile app, Signify Interact system. And you can control up to 200 zones per or 200 lights per zone. As I mentioned, it's extremely robust and suitable for wet locations. It was designed, for greenhouse applications that could be pressure washed in really harsh conditions. And then to offer even more advanced controls, we can integrate with climate computers such as Argus, Priva, GrowLink, others, and really have the most precise control of your lights and color changing, specific recipes that you could work with Fluence's staff to come up with based on what we've seen in certain research settings with certain cultivars, and we could work with you and help you understand the sensitivities and how to adjust through photo acclimation of red and intensity. So that pretty much wraps up that new product feature I wanted to highlight for you guys. Um, we're going to open it up for questions. So if you've got some questions there in the upper part of your screen, there's a Q&A tab. Open that up and fire away. We really appreciate you guys joining us today. We want to answer as many of those as we can get.
Taylor, we've lulled them into silence. This is maybe
Quiet crowd.
a first for a Fluence webinar. So, okay, we have one question. What are the characteristics of cultivars that would not be good for dynamic lighting? That's an interesting question. And personally, I don't think there are any cultivars that aren't suited for dynamic lighting. Um, again, I think there's a lot of potential that can be done one day, and so it's challenging to talk about, right? Because I'm wearing two hats here. There's Dave Hawley, plant scientist, and Dave Hawley, kind of like commercial cultivation. And those are two different entities. And Taylor really helps me speak the commercial language much better because as a plant scientist, I'd say, well, there's every cultivar has potential, right? You can think of the genetics that are underlying as what the plant could do one day if only you had the right environmental cues to turn up or turn down certain dials. And that's where we see things like, maybe there is room to do things with blue or UV or cyan or green or amber or on and on and on. But in terms of commercial application, I'd say, well, again, at the intensities that we talk about, really it's just white and red that seem to matter right now to drive efficacy and maintain quality and yield. And every cultivar out there is going to benefit from a dynamic light because even cultivars that are completely red tolerant, it's like, well, great, ramp your red all the way up and just leave it there for that particular cultivar and enjoy high yields while also having the lowest possible OPEX for that spectrum. Conversely, you may have a cultivar that's really, really sensitive to red light. Okay, well, ramp it down. There's there's no reason why you wouldn't do that. So, to the question, what are the characteristics of cultivars that would not be good for dynamic lighting? I don't think there are any cultivars that don't benefit from this. Taylor, what do you think about that?
Yeah, no, I mean, I agree. I think as we've shown, there's certain cultivars that are more sensitive to red light. So obviously you're going to have to be considerate of that. And that's where the dynamic zoning can come into play. You can create a higher light, let's say intensity zone or a higher red content zone on one side of your room than the other and run multiple cultivars in the same space after you've had time to kind of identify um, their susceptibility to photo bleaching, for example, or if their like of high light intensity or lack thereof. Like there's some genetics that just don't prefer super high light intensity. So yeah, I mean those things definitely need to be considered. But I do think there is a place for dynamic lighting with all genetics. You just got to figure it out with your own with what you've got. I see another one here, a question that came in. It was about CO2 and other variables in the indoor growing environment. How does it work? Like, I guess the tunable spectrum is what he's asking. Do you run the rooms the same way? And it's a great question. I mean, uh, in at a high level, yes, you would run it the same way, but you're going to be monitoring if you're using higher light intensity and pushing the crop harder. If that's what your goal is, then you're going to have to monitor water, nutrient uptake, CO2 uptake, and adjust accordingly so you can keep kind of a similar target EC, water content, temperature, humidity, and CO2 level. If you're pushing really high light intensity up to 1500, like Dave mentioned, or even higher, I don't see a much more need to going over somewhere around 1400 ppm of CO2. But if you're running a lower level, you definitely want to increase up to about 1400 with high light intensity.
Yeah, I would build on that. So, as it pertains to dynamic spectrum specifically, um, as far as we know, there's not a really big difference in CO2 uptake, whether you're using a more white spectrum versus like the higher red, like 80% red. It's really just a factor of light intensity that changes your CO2 consumption. So it's a really, really good question to ask. But as long as you're running sufficient CO2 for the overall light intensity that you're working with, you should be okay whether you're using more white or more red. A couple more questions in here. So I'll answer the easy one first. Someone asks, when will the Raptor 2 be available for ordering? Q4 this year. So sometime later this year. Stay tuned. Next question. Why does the Emerson Enhancement Effect not work on high PPFD cannabis? Great question. It probably does. It's just that high PPFD for cannabis as a biological system is really, really high. Like higher even than the highest commercial cultivation is doing right now. Uh, a few years ago, back in like 2020, Taylor, you were working with us in a slightly different context at the time, but we were working on a high PPFD, like a light intensity study, right? And on that study, we took cannabis up to intensities of about 2,500 micromoles. And we thought, well, this is going to kill them. But, you got to see where the breaking point is. But it didn't. Um, we had three different cultivars, and what we saw was that for two of those cultivars, we were having a little bit of a plateau in response. In other words, we weren't getting like a 1-to-1, 1% more light is 1% more yield. We weren't seeing that anymore. It was starting to plateau. But for one of the cultivars, it just kept going up linear. 1% more light give us 1% more yield. And we said, well, okay, that's that's kind of ridiculous because you'd walk into that research room and the ceiling was just lights. You could kind of see a bit of scaffold poking through that we mounted lights to, but it was like, all right, well, no one is going to do this. This is so expensive for us to even run this study. We had to move electrical capacity from other rooms into this one room just to be able to do the experiment. We'd maxed out our HVAC, and we did that, and we said, okay, well, obviously there's headroom here, but we just can't go any further. It just doesn't make sense for the business. It doesn't make sense for the commercial grower. So let's just say 2,500, that's our cap. But look, to get back to the point of the question, the Emerson enhancement effect, again, it's most impactful when you're getting to some kind of bottleneck in the photosynthetic machinery. So at that point, you have to make sure you're not having a bottleneck with HVAC or your climate. In other words, you're not having a bottleneck with CO2. And if those things are all good and smooth sailing, then you can really push the plant as hard as you can. And if you get to the point where photosystem 2 and the electron transport chain are just driving really hard and they are facing that bottleneck at photosystem 1, where you would get a boost from far red and get that Emerson enhancement effect, then it's relevant. But I think with cannabis, the point where you have to get with PPFD is just so high that it's not something that we're going to see anytime soon. It's just one of those things like, Fluence isn't going to do it because it's not relevant to our business and we can't afford to spend money on projects like that. But even in academia, it's one of those things where, thinking about the labs in the world that are doing cannabis research, it's again, they'd face the same challenges we had. They'd need a lot of light, it'd be really expensive. And it's a cool academic question, but I don't know who's going to pay for it. Unfortunately, that's kind of the thing in life, right? Everything costs money. So, long way of saying the Emerson enhancement effect could be relevant in cannabis, but just at very, very high light intensities that we're not likely to see anytime soon.
There's a good question here about lighting, dynamic lighting in a greenhouse setting. So it's from Dominique Van Grusen. What's your perspective on using dynamic lighting in greenhouse setting where you use LEDs to supplement sunlight, providing the spectrum of the sun also shifts during the time of day and time of the year? Yeah, that's a great one. I'd say that's where really, dynamic spectrum is going to be exciting to play with all the different types of recipes and computer control reactions so that depending on a DLI or seasonal changes in light intensity, and as you mentioned here, the changes of the spectrum throughout the day, we can keep either a uniform light distribution if that's what we want, or the most energy efficient. I think it gives you the flexibility of doing both. What would you say, Dave?
Yeah, I would echo all that and I would build on it a little bit. Where my mind went was that the ROI calculation, the return on the investment, is a little bit different for a greenhouse than an indoor facility. Essentially, pardon me, essentially you are getting some red photons from the sun. And yeah, those red photons are more spread out, evenly between 700 nanometers. So kind of the biophysics are a little bit different. But when we look at the ROI for dynamic lighting in a greenhouse, the ROI is not quite as good um, because there's less room to really play with the red and white range than indoor, but it is still there. So all that to say, dynamic lighting in greenhouse makes sense, particularly if you're somewhere like an Exo, Dominic, and you really in the winter you're getting like a DLI of 2 in the winter. Maybe I'm exaggerating, but the point is,
Five. Yeah, something low.
it's you're not getting a whole lot of sunlight through the winter, and so a dynamic lighting solution can really save your OpEx just as it would in a greenhouse environment. There's a couple questions here, Taylor. There's one that I see on blue and UV. We could talk about that. And then the other question is on UVB. Do you want to go first on those or shall I have at it?
Um, well, I'll do kind of, yeah, I'll start and let you bring us home. It's my understanding that UVA, UVB are very similar in their effects and how they could be utilized. UVC is very damaging, right? So it's actually going to be used for sterilization and IPM potentially purposes in very low quantities, and actually usually during the nighttime or the non-photo period time, it's when it's been shown to be the most effective treating things like powdery mildew. But as far as the question around UVB having an effect on the resilience of the plant, I think there's probably something to that, but would assume you're going to have to introduce it at a young stage, right? Propagation, seedling, and carry that through the entire crop cycle, which would allow for that resilience to kind of build up in the plant. What we saw with, especially with flowering experiments where we were looking at potency and yield impacts, it was actually too harmful at that point. And the plant, wasn't accustomed to the UV. So I think it did more harm than good. But as you mentioned, I think there's still more work to do there.
Yeah, yeah. So I really like both these questions because UV, I actually think is super exciting. And I wish that, if I didn't have so much to get through in this presentation, I would have lingered on the UV topic longer. So thank you, whoever you are, Anonymous, for bringing it up in the Q&A. UVB, you're right. In your question, you said it has an effect on the resilience of plants. Thicker leaf surface, for example, same as UVC, and the stress is systemic. Yeah. So as far as cannabis is concerned, just like Taylor said, we do believe there may be opportunity for UVB in the future, and just applying it with different doses throughout flower when it otherwise hasn't been exposed to UVB is not the way. We think there may be potential for a sort of systemic hardening of the plants if you apply UV earlier in development. And like I said, that is on the research roadmap for the not too distant future. It's a somewhat challenging study to do because you need some specialized chambers and specialized safety things and so on. So it's been on the radar for a while, and we simply haven't had the means to do it. That has recently changed with the new research facility we're going to be moving into soon. So again, UVB during vegetative development, we think could result in some sort of hardiness that benefits yield or chemistry, to be determined. There are also groups out there like a New Zealand lighting company called Biolumic that's done a lot of UV research on early seedling development. And I would encourage you to take a look at their work. Suffice to say, they have solutions for field crops where they've applied UV very early in development that have resulted in much higher yielding and hardier plants. Um, Once again, maybe there's potential in cannabis, but that works just, there's a lot of research to do still. Now, the next question had to do with how does blue compare to UVA in research? And I'm very excited. As Taylor was talking, I was rooting through my desk trying to find different scraps of paper of notes I took at ASHS recently. Someone was presenting on the effects of UVA and blue in another plant species, and they explained to me after the presentation that UVA and blue are both received by the plant by cryptochromes, but there are a lot of different types of cryptochromes. Um, there are cryptochromes that are more sensitive to longer blue, like 450, 460. There's cryptochromes that are more sensitive to shorter blue, like violet around 403, 408 kind of thing. Cryptochromes that are sensitive to UVA. And then all of these cryptochromes are more or less sensitive depending on the intensity with which they are receiving those photons. So again, I would say that's an area where my photobiological knowledge is a little bit lacking and somewhere that clearly I'm eager to learn more. So I know that wasn't the greatest answer to say, well, use UVA like this and use use blue like this. But what I will say is that the biochemistry uh, seems to be a little bit different between UVA and blue. And that's something that we're going to have to take a look at more in the future.
We had a couple more come in. I know we're running short on time, so I don't know how deep we can get. A couple really good ones, actually. Um, I think one we can tackle really quickly was the first one that came in, or the next one. Many cultivars take a lot less light than 1500 UMOL. The Blue Dream you mentioned is a really high PPFD loving cultivar. Will far red work better on those low intensity cultivars? And I think that kind of depends, right, on the genetics. What we've seen, there is at least one genetic that we've looked at that tended to respond favorably to far red, right, Dave? I want to say that was a um, pheno of Jack Herrera, I believe.
Yeah, that's right.
But yeah, but overall, we hadn't seen that much. So if you start using far red with your genetics, you may see that at lower light intensities. But and as Dave mentioned, remember if you're adding light, whatever, if it's additional far red to what you had, your base light, you should be seeing an increase in yield because you're adding more light.
Yeah. And just to build on that a little bit. So, kind of like in my part of the presentation, I was saying, here are the results of our research at 11 to 1500 micromoles, but there are all these other studies from Wageningen University and so on that have looked at spectrum at lower intensities. You know, I see that and I think, okay, that's really interesting. That probably means that at higher intensities, we've just turned every knob up to 11. And, you know, if we fiddle with the spectrum a little bit, every knob is still stuck at 11 because we're still just at the point of saturation for everything. So I think, okay, I wonder if maybe one day the world of commercial cannabis production is going to look a little different. You know, everything's going to be kind of dialed down. It's going to be kind of like this low and slow method of cannabis production. You know, a little bit like doing good barbecue, where it's like, if you just dial things back, you have more room to manipulate the chemistry with clever environmental inputs. Again, we don't have those answers right now, but it is something that is on our radar, and we think one day that may be how people go. Um, I think Taylor, with that, unfortunately, we need to wrap. Um, so I'm going to say thank you everyone for your attention. It's always a treat to present to an audience, especially when we get some good Q&A after the fact. If you would like, I see there are a couple questions we couldn't answer just because of time. Please email us. You see our contact info on screen right there. Me and Taylor will email you back. In what you can tell here is the answer to the question may be we don't know yet, but that's the nature of Fluence. We do the research and we tell you what we know and what we don't. Otherwise, thanks once again. And everybody, have a great day.
Thank you all.